This program contains graphic images and discussion of medical procedures. Viewer discretion is advised.
I'm Katrina Jameson. I go by Kat. I'm the director of the Sanford Stem Cell Institute at UC San Diego. And we're thrilled to have another mind-bending mind event, which is all about the mind. So we started these events here thanks to Catherine Canjo, allowing us to use this lovely space, the Museum of Contemporary Art, to think about medicine informing novel discoveries. How do we think differently about the art of science and medicine, and how do we find new solutions together? So the point of these mind events, how many people have been here before to a mind event? Wow. Okay. So we have repeat customers, audience, and participants. We really want your input. We are trying to find new solutions for tough diseases. And in particular, today we'll be talking about neurodegenerative disorders, spinal cord injury. When you think about stem cells, they can regenerate, they can repair tissues, and sometimes we have to replace them because the stem cells we have just don't work very well, particularly in inflammatory environments. At the Sanford Stem Cell Institute, we have 6 centers and a brain tumor and neurorestoration program. We've got Professor Kalesi here representing the brain tumor and neurorestoration program, Frank Fernari also representing the brain tumor and neurorestoration program. We've got the Sanford Stem Cell Clinical Center, represented Sheldon Morris here. And let me just see, we have the Innovation Center, the Advanced Therapy Center, the Discovery Center, and we'll have them here, the leaders here, for other events. But the point is, we're going to transform medicine together. We are not going to wait for other people to come in up with the solutions, because we live in a unique town where we have industry, academia, philanthropy all working together. And we tend to collaborate a lot. It's in our nature. It gets into your bloodstream after a while. We have a number of patient advocates in the room, and that's the way we like it. We want to get it right. So in terms of the lineup today, you'll see we have a lot of illustrious speakers and people who've really helped to shape the field. I'm particularly grateful for Larry Goldstein being here because he started us on this stem cell journey. And I still remember when Dennis Carson took me over to your office and said, you have to meet Larry, and the rest is history. We really started working together on educational programs to begin with for stem cells, and then said, wait a minute, we've got to get therapies to the clinic faster. And thanks to T. Denny Sanford and our campus leadership, we've been able to do that with our chancellor being rather innovative. So today we're going to really focus on stem cell pathways with a target on neurodegenerative disorders. We've had the good fortune to work not just locally, nationally, but also internationally. So the first person I'd like to introduce is actually Professor Jun Takahashi, who's joining us remotely from Japan. He pioneered induced pluripotent stem cell technology for Parkinson's disease. He was able to make a dopaminergic neuron product, and the PI of that trial here will be talking about that in just a few minutes. But I wanted to shift to Jun Takahashi, who is our first visiting scholar in the Division of Regenerative Medicine when we started it in 2014. So we'll go over to Jun as our first presenter. He just had a Nature paper on a Japanese study on the induced pluripotent stem cell-derived dopaminergic progenitor product 2 years after implantation of the first patient. So our friends at the back are just going to get Jun up and running. I think the point is we live in a very international place where people have to come to San Diego. So we like that. We have a lot of meetings here, but a lot of us aren't from here. So there's no, well, you're not from here, you can't be involved. We're all part of the club. So I'm happy about that. So Jun trained with Shinya Yamanaka. Does anybody know what Shinya Yamanaka got the Nobel Prize for? Induced pluripotent stem cell technology, 2012. So this is the trial that came from that. Hello, Jun. How are you?
Yeah, I'm very happy. Thank you. And also, I'm sorry, so I cannot attend on site.
Oh, yes. We're always happy when you're in town, Jun. But if you can go through your few slides. Everybody today is limited to 5 minutes unless we want to stay longer. So I realize it's very short, but I know as a neurosurgeon, you can also be quite quick.
Yes, I'm Jun Takahashi. Hello, everyone. So from Saira, Kyoto University. And so first, I would like to thank Professor Jamieson for this wonderful opportunity. And by the way, so I was working in the Salk Institute Rusty Gage's lab 30 years ago. So this is a picture at that time. So, but anyway, so today I'm going to talk about UCSD clinical trial for Parkinson's disease patients. It is transplantation of human iPS cell-derived dopaminergic neurons. This is collaborative work between UC San Diego and Kyoto University and Sumitomo Pharma. So this is a Japanese pharmaceutical company. We started this project in 2016. So finally, so we could start the transplantation this year. So I'm very glad of that. First, Parkinson's disease is caused by loss of dopaminergic neurons in the brain, and it causes the motor symptoms like tremor or loss of movement. So that is a reason why we want to replace the lost dopaminergic neurons by cell transplantation. To make dopaminergic neurons, we used induced pluripotent stem cell, iPS cells. iPS cells can proliferate unlimitedly, so we can have the large amount of cells. And also, iPS cells can differentiate or change into a variety of cells like neurons or muscle or other type of organs. Theoretically, iPS cells can change any type of cells in our body. This is the outline of this trial. Sumitomo Pharma make dopaminergic neurons from iPS cells in Osaka, and the cells were transferred to San Diego, and neurosurgeons transplant the cells, and the neurologists and radiologists evaluate the safety and efficacy. The same trial has already conducted in Kyoto, Japan, from 2018 to 2023 with 7 patients and 2 years observation. So we could confirm that iPS cell-derived dopaminergic neurons are safe. That means no severe adverse events and no tumor formation. And also functional. It means grafted cells survived and produced dopamine in the brain. And effective. The cells improved the motor symptoms of the patient. And we reported this result in Nature 2000 this year. I will show some data. So this image shows the dopamine synthesis in the brain before and 1 year or 2 years after cell transplantation. So as you can see here, this area shows yellow or red, green color. So that means the dopamine synthesis is reduced in this area. And we grafted the cells in this area. And after cell transplantation, this area turned red color. So that means the grafted cells survived and produced dopamine in this area. How about the symptoms? The symptoms of the Parkinson's disease is divided into 5 stages. Grade 5 is most severe stage. As you can see here, at least 4 patients out of 6 shows the improvement of these symptoms. So very severe patients may not respond very well, but anyway, so we could confirm the informed improvement of the symptoms. So this UCSD trial, it is to confirm the safety and efficacy of the cells in the United States. And if we could confirm both in Japan and the United States, it gives strong evidence for this therapy. And it also provides an opportunity to treat patients with Parkinson's disease in the United States. So that's all. Thank you for your attention.
Okay, thank you so much, June. So we have friends in many places, but June is a major patient advocate and pioneer here. Thank you for being here. Next, we'll turn to Dr. Joseph Chachi. And we started our first trial with Dr. Chachi in neurosurgery with the most impossible target. And Larry, you remember this, that we just started the Sanford Stem Cell Clinical Center back in 2014. We said, Dr. Chachi, how would you like to treat people with spinal cord injury? He goes, of course I can do that. No problem. I can practically do it in my sleep. So now we're going to hear about the circadian rhythm disruption for Dr. Chachi. Thank you for being here. He's an extremely gifted professor of neurosurgery in the Department of Neurosurgery. We've got Alex Kalesi here representing the Department of Neurosurgery as chair. This is the first in the world treatment that Dr. Chachi is going to tell you about today that started 10 years ago. Thank you for being here.
All right, good evening, friends. My pleasure to be here. I'm Dr. Joseph Chachi. I'm going to share some brief remarks about some of our work in spinal cord injury here at UC San Diego. Many of you are aware how devastating in evolution spinal cord injury is. What hits really hard is that it's frequently young, healthy, active people who end up in what they perceive to be a hopeless situation. And so that begs us to work on something like this. And why is it difficult to treat spinal cord injury? Well, many reasons beyond the technical challenges are the fact that the spinal cord doesn't regenerate. And so the body forms a cavity and other things happen with inflammatory responses that sort of block the growth of the regeneration of the spine. So we've published on these challenges and figuring out ways to sort of improve the options. And what are some of the current treatments? Rehabilitation, neuromodulation, we've studied that and published in that space, but we're here to talk about stem cells. Why are stem cells cool? Well, they're cool because if you do it right, and we're working on that, you can get them to become whatever you want them to be and whatever you need them to be. And in the spinal cord, we're trying to fill that gap and create a bridge to allow that regeneration to occur across the level of the injury. So how does one get to do a first-in-human clinical trial? Well, candidly, it really couldn't happen any other place but UC San Diego. And in collaboration with the Department of Neurosurgery, the Sanford Stem Cell Institute, and all of the brilliant minds that work with us in this space, upon whose shoulders we stand to look into the future. And I can't hesitate to point out the bravery of the patients, because imagine the bravery that it takes to be the first human in a first-in-human clinical trial. That's a brave individual. So this is 30 seconds to summarize 10 plus years of work. We did large animal studies, demonstrated grafting of the cells and safety, developed and modified devices, including creation of floating cannula and magnetic needle in order to deliver the stem cells into the spinal cord. This is a video that gives sort of an overview of what one of the patients looks like. That's the imaging of a cervical spine injury. This is the platform that we developed. This is a view under the operating microscope with microdissection techniques to access the spinal cord parenchyma. This is an actual stem cell transplantation injection with the floating cannula as part of our first clinical trial. And you can see that we did multiple injections in the site of the injury. This is another video sharing what the platform looks like in place in the patient, what the stereotactic delivery components of the platform are like, and again, sharing the injection. I have to show this a little bit to show my neurosurgical colleagues that microdissection technique does exist in the human spine, and that we are capable, some of us. So we did 2 studies with that device and cervical and thoracic, and we published a couple papers on that. Anyone who's interested in more granular detail can find me. I'm easy to find. I'm everywhere. But I want to share that there were no adverse events. none at all. And everything went well despite immunosuppression that was required for the study, despite the risks of revision spinal surgery. And so that allowed us to move forward to the study that we're doing, which is currently enrolling, which is also a first-in-human stem cell transplantation for spinal cord injury, testing a patient-mounted delivery device that we created and doing intraparenchymal injections of a cell line called OPC1. These are some intraoperative photos sharing what the transplantation looks like. You can see the device. This is a magnetic needle, which also allows the patient respirations and pulsation of the spinal cord to continue throughout the injection, as opposed to previous studies which required holding respiration. Intraop video, just demonstrating similar injection technique, but a new device in a thoracic spinal cord injury patient who was the first human enrolled in this first-in-human study. Just some quick videos about what's in the pipeline for hopefully next clinical trials. This is one of those, which is a sub-peel injection to deliver stem cells. And it's a pretty neat technique, and we've created devices for that too. I'll play these videos simultaneously to respect Dr. Jamison's schedule for the cocktail hour. She's tough, I got to tell you. She is. But you can see the technique of putting the needle in the sub-peel space, which not many people thought was accessible, and then you can see the dextran blue spreading in that space. And actually, we've been really happy with some of the preliminary animal studies. As I mentioned, it's a collaboration. Dr. Klessy's here, Dr. Jamison's here. I don't know why I'm here. Dr. Masala's not here. Many other collaborators. They don't all fit into a phone booth, but here's a picture of the study team. Not all of it. It's a huge study team, and I'm grateful to every one of these people. And, you know, in anticipation of passing the baton to my colleague, partner, and really good friend to talk about the Parkinson study, this is a picture of Dr. Takahashi actually in San Diego. So he is a real person and has done amazing stuff. And he and Dr. Benheim are the big thinkers, and I'm just standing there nodding my head, which I do very well. And I think I'm going to let the robot introduce the next talk. Ready? Thank you. Dr. Duval.
No, it's perfect.
All right.
As always. Yes, he's definitely the brains behind the operation for how we got started in this field and how we've expanded to other sites in the regenerative space when it comes to replacement therapy, including designing this device. So thanks, Dr. Chachi, for that. We also have the editor in the room that, remember, was responsible. Dr. Chari, Sheila Chari is here. She gave us a chance when other people said, this will never work. And yeah, Dr. Chachi made a very strong pitch for why it matters. And these patients are doing well. So fabulous. No adverse events. Absolute neurosurgical prowess. Also, I would say we have the Sanford Stem Cell Clinical Center, our outpatient and inpatient service where we can help with people. I think for the next part, we're going to pivot to something, it's hard to follow Dr. Chachi, but something a little newer that you were hearing from our friend Dr. Takahashi. So we're going to move to Sharona Benheim. So Dr. Benheim is a specialist also in replacement therapies when it comes to neurodegenerative disorders. This is in the brain this time. So she has a lot of experience in terms of neurodegenerative disorders like Parkinson's disease. When Larry and I went to Kyoto in 2014, not that we're keeping track of time, we couldn't imagine a better team that would be added to Dr. Chachi and our neurosurgical team here. And then Dr. Benheim came forward and said, I'll be the PI of this trial. There's a lot going on in the Parkinson's disease space. This affects 1.2 million people in this country as a diagnosis. And there's a quite substantial amount of neuroinflammation. So it means your surgical techniques have to be perfect. So we're thrilled that this trial came here. There are a lot of people in this room that helped with this. We have the Sanford Surgical Training center to help, but nothing replaces the best neurosurgical team in the world. And I know I keep talking about Alex Kalesi, but thank you for making sure that everybody thrives here for our Sanford Stem Cell Clinical Center as well. So I'll introduce Dr. Benheim. Thank you for being here. She's also the PI of the Neurona study that has had NBC News. So thanks for being here.
Thanks for that very kind introduction. It is such a pleasure to be here with you all. I'm Shrona Benheim. I am a functional neurosurgeon here at UC San Diego. And just to take us back a step, you know, what really is cell therapy? A lot of you have been to these events before, so you have a greater understanding of sort of the bigger picture here with our fantastic leaders really leading this space coming from San Diego. So really briefly, what are we talking about here? It's really an innovation that involves the transfer of specific cell types into a person to treat or prevent a disease. And it really is such an exciting new wave of therapy that came on the heels of a ton of innovation over the last several decades, really spearheaded by people like Jun Takahashi, who you heard from today, and Yamanaka, his mentor, and the people here, right here in San Diego. And it really allows us, these techniques that have come about in the last couple of decades have really allowed us to more easily create specific cell types by harnessing and controlling really the power of stem cells. And so, a lot of work has been done in different types of cell-based disorders by injecting cells directly into the cerebrospinal fluid or by injecting cells through an IV into the blood. And so what we'll talk about today is what we do in the Department of Neurosurgery at UC San Diego in new innovations in cell therapy, where we're actually injecting these cells directly into the brain, usually into specific regions where they belong. And I'll take you inside of our operating rooms a little bit in this talk and show you just how we do this. So a trial that I won't spend much time talking about today, but one that Dr. Jameson alluded to is a trial actually to treat epilepsy, which I'm really excited about. It's one where we're injecting what we call GABAergic cells that actually promote inhibition into the hippocampus of patients who have refractory epilepsy. And by doing this, we're hoping to restore this balance of inhibition and excitation in an area of the brain where we know there's too much excitation by actually injecting these cells to try to calm those regions down. And currently, our only options for these patients are actually either to remove that part of the brain or ablate it. And so this really is a completely revolutionary new way of thinking about how to treat epilepsy by actually restoring some balance, by correcting the underlying pathology. And it provides options for patients currently who have epilepsy who really did not have any good treatment options before these types of therapies came out. So it really opens up our ability to treat patients who aren't eligible for some of the therapies that we have currently. So very excited about this. We're able to perform these kinds of cells. This is an example of actually being able to see the real-time injections. Those little tiny white dots kind of look like beads on this MRI. We're able to do this in part because we have some really cool advanced technology, which I'll highlight today, including our intraoperative MRI here. And this is right over the hill at the Jacobs Medical Center. We actually have an MRI that lives inside of our operating room, and you see here, it actually comes out on rails onto the patient so that we're actually able to visualize in real time what we're doing. We use this, of course, clinically in some of our epilepsy cases and other types of functional neurosurgery and even tumor cases, but really gives us an advantage in some of these cell therapy trials where we're really able to very precisely visualize exactly where we're depositing these cells. So really great technology that allows us to be a part of some of these advances. So I'll switch gear now and talk about cell therapy for Parkinson's disease. We're a part of 2 clinical trials at UCSD for Parkinson's disease, very similar with some slight nuances. And you can see here, these are actually patients whose brains we've reconstructed. And these this green outline is actually the area that we're targeting in the brain of Parkinson's patients called the putamen. This is an area where we know that dopaminergic cells are lacking, as you saw in Dr. Takahashi's presentation a little bit earlier. Some of the differences with these trials are actually, one of the trials, the Aspen trial, takes a patient's own skin cells, then reverse engineers them into stem cells, and then allows us to create dopaminergic cells from that, and then we inject those young dopaminergic cells into the brain. And another version of this, we're actually taking those cells from a cell line and then reverse engineering them into dopaminergic cells and injecting those cells. So very similar approaches with some slight nuances that allow us to use technology that just previously didn't exist in order to treat these patients. So it really takes a very large team to take this on, and really this just represents a small part of our neurosurgical team. Dr. Joseph Chachi, who you heard from, is the PI of this trial, and it's really been a pleasure to work with him on this particular trial. We have some very talented residents who are working with us as well, Kiefer Forseth and Mickey Abraham, and of course this is all under the guidance and leadership of the chairman of our department, Dr. Alex Kalesi, who's been just an incredibly supportive partner in allowing this to expand this technology. And of course, this team includes our OR staff, our cell lab personnel, technology specialists, but really the Stem Cell Institute led by Dr. Jameson is really what allows us to be able to coordinate this massive effort. So Dr. Takahashi already talked about this. I won't belabor this so we can all get to the cocktail hour. But we know that we are treating patients who have the clinical signs of Parkinson's disease. We have some very rigid inclusion criteria because we want this to be a very scientifically rigorous study. Patients have Parkinson's for 5 years and have an inadequate response to current therapies, specifically drug treatments. We have an age range, and then we have some really specific criteria that Dr. Takahashi already alluded to about who might be a good candidate, at what stage of Parkinson's they might be a good candidate. You're not expected to actually read this slide, but this really kind of shows you the level of coordination necessary to perform just one of these cases. And it starts from well before we get into the operating room, doing very specific imaging studies, including F-DOPA scans, which is a very, very unique way to see dopaminergic activity in the brain. And then leading up to the operating room, there's a very specific way we prepare these cells that has to be done in real time. A lot of this preparation is done in Japan. They're then flown to the United States, and then more preparation is done in our own cell labs as well as in the operating room, and then injecting the cells. Here's a picture actually from just last week where we performed one of these cases. You can see here our clinical team, our intraoperative cell lab prep team, and a lot of our ancillary personnel who were all there to make this happen. And you already saw a little bit about the robot that we use. We actually performed this procedure under direct robotic guidance. You can see here the patient is prepped and draped. Actually, the big circle is the CT scanner that also helps us guide our trajectory. We have an MRI machine right next to it. And then we have a robotic arm that takes us to our trajectory, which is really important in this case because we're injecting 10 million cells through 56 different deposits through 3 different trajectories on both sides of the head. So it really takes us quite a bit of coordination to ensure that these deposits are precisely injected into exactly the regions that we want them to be injected. And here you can see the actual injection procedure. They're a specific, a very unique special cell therapy injection needle was actually created and FDA approved for this specific trial. And so you can see this needle here, really made by Jun Takahashi and his team, and in conjunction with our team here in San Diego. And so you can see here the method of injection of these cells in one specific trajectory. And then, of course, what we all care about are the clinical outcomes. We've already had several patients come through at UC San Diego, and we've just recently had a publication in Nature from Jun Takahashi and his team about some of these results. You saw some of these pictures where previously there was no dopaminergic activity. After 12 months after injection, we see very clear dopaminergic activity that persists at 24 months. So we're able to inject these cells, we're able to do so safely, and they are producing dopamine, which is all very exciting. And out of the 7 patients, 6 patients underwent efficacy evaluations, and we do see 20% improvement in their off scores, and additionally a 35% improvement in their on scores. So we really see that these patients are benefiting from this therapy even in these early stages. So really exciting times ahead. Thank you guys for having me.
All right. So you see, that's pretty impressive. We are actually leading the world so far in the types and depth and breadth of trials we're doing in this kind of neurodegenerative disease space because we have the infrastructure, the people power to do it. And we never thought, why shouldn't we, right, Joe? Why not do it? So now we're saying, now we want perfection. We're starting to see real sustainable effects. We heard about Sumitomo and Aspen. We've got Jean Loring, whose therapy was developed here as well. So thank you for being here, Jean. But how do we perfect things? And what about the role of inflammation in neurodegenerative disorders, where we know inflammation dampens the capacity of endogenous repair and regeneration by stem cells? I trained as a bone marrow transplanter, so I'm more surgical, but there's a medical side to things. How do we improve outcomes for people with neuroinflammatory disorders? And do the cells that we give other people's cells that are going to stop inflammation find their own way? So we're going to hear from a leader in the field, Jennifer Graves, who's leading all of our efforts for multiple sclerosis. So this is a neuroinflammatory disorder we never imagined we'd be applying stem cell therapies to. I shouldn't say we, I didn't imagine it. Of course, Jennifer did, Dr. Graves did, and she is absolutely ahead of the curve. But I've got to say a big thank you to our patients, because our patients are our partner scientists who are helping us like astronauts do for experiments in space. You're taking on the risk and the potential reward, but you're leading the charge. So thank you, Dr. Graves, for being here.
It is so fantastic to be able to join you all here at this MIND event. I'm going to get through 5,000 years and the 10 minutes I've been allotted, okay? Okay, so I thought in this beautiful space, it would be appropriate to start with images rendered by patients living with the disease about what it's like to have multiple sclerosis and Parkinson's disease. So I'll just let you take a moment to appreciate these patient renderings of what it's like to live with these disorders. My overview is you've heard some great discussion about Parkinson's disease, spinal cord injury. I'm going to introduce or reintroduce you to multiple sclerosis, what we've accomplished so far, but what are our biggest remaining gaps, and how do cell therapies potentially fill those gaps? What are we most excited about, and what is on the horizon for research and innovation? So MS is an autoimmune disorder that's triggered by genetic and environmental factors. We know the genetic component is 25% because if you have identical twins, one has MS, the other one has a lifetime risk of 25%. It's characterized by stripping of the myelin protective coating of nerves with 3 targets. So this disease goes after brain and spinal cord. And as you can see, the brain on the bottom left, typical MS lesions, the orange arrows in the middle highlight spinal cord lesions. And on the right-hand side, it also goes after those optic nerves that connect your eyeballs to your brain. And some lesions there are highlighted with the white arrows. So here's where I'm going to start off. 5,000 years ago is the origin of MS. With new genetic technology, we now know that MS probably evolved when in the maps on the bottom left-hand side, people in Europe started gathering together, bringing together flocks of different animals, living in villages, and this brought on the advent of a number of viruses, including the Epstein Barr virus. You may recognize it as being the cause of infectious mononucleosis. It's also a cause of several cancers, but it is the virus required to have MS. And now 5,000 years later, we're seeing the development of a vaccine for this virus that causes MS. And recently, our UCSD MS Center collaborated with the Sanford Stem Cell Clinic in a cell-based therapy against EBV-infected immune cells. So the gold and blue cells represent these sick B cells that have been infected with EBV, seem to cause havoc in this autoimmune disorder, and we delivered a treatment, T-cell treatment, that went and killed these B cells. And that was, I'm proud to say, our first collaboration. Some other things that are relevant for you to know, you may know someone who's lived with MS, but you may not know that kids get MS too. In addition to being an adult doctor and a researcher, I founded our Rady Children's Pediatric MS Center in the south part of the county. Pediatric MS occurs in about 5% of cases. Again, in this beautiful art center, I was showing off some of those kids' artwork. It's the earliest form of disease. It's the most active. Kids have 3 to 5 times more attacks than adults do, and they're teaching us about the causes of MS, not just EBV, but chemicals in our environment, genetic load, and there are excellent opportunities for cell-based therapies for children. You may know that veterans do get multiple sclerosis, but you may not know they have a 3 to 5 times higher rate of developing MS, and they're teaching us about the causes of MS. In fact, they taught us about EBV, and they taught us because young men and women signing up for the military have their blood drawn at 18, and we can go back in time and try to pinpoint the exact origin of MS. But they do tend to get severe MS. And recently, I had a 45-year-old Marine veteran who was breaking through all of our possible therapies, and we put him through an autologous stem cell transplant, finally got him back to his kids and his family. You may or may not know MS has a highly varied phenotype. It's a very heterogeneous disease. Everybody's story is a little bit different. And there are people with more benign and more malignant forms. And there are relapsing forms where they attack and then they remit, and there are progressive forms. The typical picture on the right-hand side shows clinical disability in the dark black line, the first upward deflections, the first attack of MS. The arrows at the bottom represent the MRI activity happening under the surface, 10-to-1 ratio of what's happening silently versus what patients are noticing, but it catches up with them later. And then brain volume, they're losing brain volume over time at the top. I thought when I was first training 20 years ago that the most important time variable on the x-axis was disease duration, but it's actually age. And relapsing remitting MS, which is all we see in kids, is a youthful expression of the disease, is currently treatable, but the progressive forms are aging-related, neurodegenerative, and they're currently largely untreatable, and that's where our mission is. So our success story in treating MS, which I think gives us hope for one day disease modification in a disease like Parkinson's as well, was based on being able to see disease activity in the upper left-hand side, highlighted with the yellow arrows, or new lesions forming in the brain from MS. And being able to target this, and the bullet points I have on the right-hand side, beginning in the 1990s, allowed us to run clinical trials. We have over 25 treatments now in 30 years for MS, where it was untreatable 30 years ago. But we've pushed it as far as we can here because we're not actually getting therapies in the brain. This is our opportunity with cell-based therapies. So this is just showing you this difference between relapsing biology and progressive biology related to age. On the x-axis is age. My kids and young adults on the far left-hand side have a lot of relapses. Some kids twice a year hitting their brainstem, high-value real estate in the brain. And older adults stop relapsing, as you can see on the right-hand side. And then the problem is those 25 therapies that we're so excited about, they're targeted against that relapse biology, and treatment efficacies decline with aging. So age is on the x-axis. Each circle represents a clinical trial of a traditional type therapy, the different colors, different drugs, the size of the circles, the size of the trials. And this meta-analysis from the NIH predicts that the medicines I have available commercially have zero efficacy beyond 53 years of age. And the problem with this is this, that due to those medications, people with MS are living longer, and due to overall improvements in healthcare. This graphic is showing you ages on the x-axis, that the mean age of those currently living with MS has shifted, and almost half will be over 55, but we have a huge treatment gap. But the good news for those of us, because a few of us are going to escape aging, is that we can try to identify someone's biological age. And this is something my group has been doing for some time. We typically divide lifespan into early life, youth on the green on the far left, later life in purple on the right, midlife in the middle. And if you drop your eyes down to the blue box at the bottom, you can measure any one individual's, including yours, I've done it to myself several times, the pace of aging to see if you're on track with your birth date, you're accelerated, or you're slowed. This construct has been linked to multiple diseases and changes in organ systems. And in MS, as I've been alluding, it's incredibly important. And the far left, with the young people, we need aggressive immune suppression. We can use sledgehammers. They tend to tolerate it, and they need it. On the right hand, we need more specific immune suppression. We need age reversal. We need the concepts that are linked to stem cell biology. And then in the middle, this is where we've been spending a lot of time measuring someone's biological age to figure out which type of biology they're likely experiencing with MS. And we've been putting our money down for a decade that looking at biological aging, both normal and expected, and disease accelerated are drivers of the untreatable progressive form of MS, and also for Parkinson's and Alzheimer's. And in this simple cartoon on the left-hand side where the blue dotted line is, if we can drive people into a slower aging trajectory, the geroscience hypothesis is we could ameliorate diseases of aging, of which all three of these are. And this, if you don't recognize it, it's Ponce de Leon's Fountain of Youth. We don't necessarily need the entire thing. We have other ways to handle wrinkles and gray hair. But if we could keep people's immune systems and their brains in a more youthful state, these diseases may be more treatable. So this is where I find out if there's any Top Gun or Maverick fans in the audience. Does anybody know what this plane was called in that movie? Oh, I don't have any fans. So this is Dark Star. This is Maverick Tom Cruise here breaking the sound barrier 10-fold over, going Mach 10. And what we need to do in MS therapies, but also the other neurodegenerative diseases, Parkinson's, as you've already been hearing about with my surgical colleagues earlier tonight, we need to cross the blood-brain barrier to get better treatments for these diseases. And here's another collaboration that we have ongoing with the Sanford Stem Cell Institute. That is to deliver cell-based therapies that can break that barrier and get it where MS is attacking the brain and spinal cord. So in this simple cartoon on the left-hand side, the blue cells are engineered CAR T cells, immune cells that we can take off the shelf, get them across the blood-brain barrier, and go after those B cells. Remember them from a couple slides ago? These are the ones harboring EBV. These are the ones having abnormal immune responses and causing the damage. And unlike some of our other treatments that we're using in MS, like rituximab and ocrelizumab, they're actually getting to the target. So what's next for treatment? What's on our horizon? What are we excited about? Well, we want to hack the biological clock. For all of us, we'd like to turn back time, but particularly for patients with these diseases, because their age is driving this neurodegeneration. And we want to harness the power of cell-based therapies and combine it with rehabilitation and nerve stimulation. But future treatments need enhanced targets. If I can't measure the treatment effect, it's going to be hard to judge whether or not these novel therapies are working. So as another aspect of our work, now some of you will know who that is. That's Charcot, one of the gentlemen who was credited with the neurologic exam. And I think, biased as a neurologist, the neurologic exam is the most elegant of medicine. But these tools in my pocket were invented in 1888 and 1889. So for neurologists, we've needed an upgrade. And here's where I find out if we have any Star Trek fans in the audience. We need something like what Dr. Crusher has. Now, I have a feeling we might have a Star Trek fan. Does anybody know what this is called? Yes. So we need a Dr. Crusher tricorder. So part of my group, we've been working on neurological engineering along with other colleagues in neurosciences. So we are creating those 21st century tools so we can measure outcomes in these trials. Now, here's a set of studies we did. You don't have to digest everything happening on this slide. Just notice that the bracelet in the middle is a wearable multi-sensor. It has 14 channels of data, and you didn't know you were going to do this tonight, but you're all going to become signal processing engineers with your eyes with me. We just had patients with MS tap their fingers 20 times and their foot 20 times, and if you look in the upper right-hand side, that multicolored tracing, that's someone without disability tapping their foot. The third tracing down is somebody who does have disability, and you can see with your eye the signal change, but mathematically, we can extract a lot of features out and judge how people are doing. Now, here's another fighter pilot photo on the right-hand side. We're also using eye tracking to monitor disease in MS and Parkinson's and Alzheimer's. A pilot has to jump their eyes across a bunch of places in the cockpit. For people with neurologic disease, this is very difficult for them to do. A quarter of your brain is dedicated to that, so we are monitoring that. And lastly, we are using electrophysiological biomarkers of cognition in multiple sclerosis and Parkinson as outcome measures. So we can put disposable electrodes on, and again, you're going to be a signal processing engineer, and you're going to look at the difference between the red curve and the blue curve and the colors on the top of the head, where we can actually map out where the differences are in thinking in these diseases. So in summary, our mission and our opportunity is that we've come a long way through innovation and research. We had no treatments for MS in 1990. Now we have 25 plus. I think we can repeat this in diseases like Parkinson's, which turn out to probably have early immune origins 20 years before we think the disease started. We need to treat to target early the pathology of diseases like MS and Parkinson's that lies beneath the surface, and daily is taking away the freedoms of walking, thinking, and having control over the body with these 21st century approaches. And cell-based therapies can actually get into the brain and spinal cord, as we've been discussing, and these are the targets of neurodegenerative diseases. This is not me, but it's a biologically younger version of my DNA. Very grateful to all of our team and collaborators. And I'll be very happy to take questions as a panel or later on in the evening. And now it is my privilege to introduce our patient advocate guest for the evening, who not only is someone who elegantly and with grit and grace lives with multiple sclerosis, but is a leader in the field of healthcare. He holds a MBA from Columbia University, a BA in human biology from Stanford. He's renowned for his contributions in the venture world. He's been on the board of more than 20 public and private companies. In 2009, Grant received the Lifetime Achievement Award from the National Venture Capital Association, and I couldn't be more pleased to introduce him and have him share his viewpoint as a person living with MS.
Thank you very much, Dr. Jamison and Dr. Graves. As I watched your slides, I think back on my own journey, and I've lived through a lot of those twists and turns that you talked about. And I'll comment on that later, but I'd like to start off with a short reading, an excerpt. But first, I want to make sure you don't think I'm a snoopy kind of person. But this story unfolded in my kitchen, and I was walking around one day, and I happened to see a nicely typed paper sitting out in the kitchen, and I glanced down at it, of course, curious what it was, and it was titled "A Slow Death." And why I think this is relevant is I think it tells you something about MS and living with MS, but it also tells you about the profound impact MS has, not just on the patient, but on their families, on their colleagues and whatnot. So these diseases go way beyond the immediate impact on the patient. The slow death. My dad has multiple sclerosis, a disease that slowly eats away at his central nervous system, turning his immune system against itself and destroying his body. In 7th grade, he told my brother and I that he had been diagnosed with MS. But the idea of my dad being attacked by some invisible enemy wasn't real to me. As a 12-year-old, I couldn't relate to what he was saying. However, throughout high school, his physical deterioration has become more and more apparent. First, he stopped skiing, instead opting to drive us back and forth to the mountain. Next, he stopped running, but he needed a little help walking long distances, just a shoulder to rest his hand on. But still, he remained strong, defiant of the disease, with the firm belief that he would be okay in the end, that MS could and would be beaten. But then came the falls. the accompanying thud, and the yell of pain. And I finally realized that the diagnosis of MS really meant it wouldn't let go of my dad. It would pull him down to a wheelchair. And the fear of that weighs on my heart every day. And that was part of my son's application to college. And again, I think it's relevant because it captures some of the human side of this disease and what it means and where it can go. What I'd like to do is wind the clock back to when I first got diagnosed. I have a few general observations I'd like to make, and they're marked in time by one point back in 1998 when I was formally diagnosed to the current time we have today. And there are a few general observations. This is really kind of an awful disease. And if you go back to the days of 1998, the outlooks were very, very bleak. A lot has changed since then. And the good news is there's been an evolution of the research, the understanding, the drugs, the treatment, and we should celebrate that. However, my third observation that I want to make is there's still a lot more to do. As Dr. Graves pointed out, we still don't understand the basic mechanisms that trigger the disease. 25%, as she said, is certainly attributable to genetic predisposition factors, but it takes the right kind of catalyst externally to really drive that home. So if I reflect back to 1998 and the diagnosis, I'd been doing a lot of running. I love physical activity outside, and I kept pulling a muscle in my leg and thinking, there's something wrong with my back. I've got to figure this out. There's a nerve problem in my back. Well, I eventually ended up with a neurologist and went through a series of tests and lots of questions and probing and MRIs and whatnot. At what point he asked me, Grant, have you ever felt this sensation of numbness or tingling? And I sort of scratched my head. I said, I don't know, but yeah, as a matter of fact, if we go back to like 1982, I'd wake up in the morning, I'd go for a run. My whole left side of my chest would be numb. My left side of my arm felt like it slept on it all night long. It was numb and it was tingling. And he goes, aha, very interesting. So he ultimately said, Grant, you have MS. And at that point in time, there were no approved drugs in the United States to treat MS. The prognosis was very bleak. The descriptors were quite depressing. Progressively degenerative disease for which we have no cure or viable treatment. In cases, we use steroids to help knock down inflammation, and that's helpful, but it's not curative in any way, shape, or form. So let's jump ahead to today. So during that period of time, I experimented with a couple of different drugs that eventually were approved. One was a beta interferon called Avonex, and I don't think it helped me at all. I think it only made me feel sick half the time whenever I took it. The second drug was a B-cell ablation drug, which Jennifer referred to, which interacts with the Epstein Barr virus. And I think that was quite helpful. But the good news is I'm really excited about this. 2 months ago, I was visiting with my neurologist up here in Northern California. And after we went through our evaluation, he goes, Grant, this is really great news. You are disease stable. There is no evidence of active disease in your body, and we should consider stopping drug treatment of the disease-modifying drug called rituximab, or this is ocrelizumab. It's in that same family of anti-CD20 drugs. So I've stopped the drug. So I think if I end my talk right now, the answer to what's happened in this period of time is nothing short of extraordinary, and I'm thrilled about it. The other thing that I've had the privilege to do during this period of time is just out of curiosity and sort of my background, things I've been involved with. I've been drawn to a lot of the research initiatives in the field. I was involved and felt privileged to be able to help out on the early stem cell funding initiative in the state of California. I helped somebody put together their original research plan for what's called the Myelin Repair Foundation. And that's been great. I've kept an eye on this stem cell therapies, which is exciting to hear more about again today. And most recently, I've been involved with a precision medicine company that looks at new ways to look at the molecular basis of disease activity in MS and give new tools to physicians and patients to understand how the disease is evolving and how it's reacting to things. So it's been, for me, a real privilege. The two key things I focused on during the course of my MS as a patient, as a person, one was noticing the impact physical activity can have on a disease. And I had a profound story. I was up having my MRI examination, and as we heard earlier, the brain shrinks as we get older. It's measured in these MRI studies I've been in for 19 years. It shrinks even more with the interjection of MS. At the end of my visit up at the MRI center, the doctor comes in and says, "Grant, what have you been doing?" I said, "Well, what do you mean, what have I been doing?" He says, "Well, we thought we had a software artifact last year because your brain didn't shrink at all, it actually grew. And we thought it was a software artifact, so we, of course, don't want to get your excitement up about that. But it happened again this year. What's going on?" So I started beating on the table saying, "Well, with my scientific prowess, no way I'll ever get in Nature magazine, but this is the opportunity. We got to get a cover story about this." So we laugh. The doctor's very serious. "What are you doing?" The key to that whole thing is as much exercise like you could possibly do, because MS is a disease like many others where you're fighting atrophy. You're fighting the natural wasting that goes on in your body, and it's accelerated by these other factors. So that to me has been a profound thing. I try to exercise 6 days a week. I try to do 65 minutes on an elliptical trainer 3 days a week, and then I do resistance weight training. So I'm as active as I can be and still remain safe in movement throughout the rest of the day. So the long and short is there are a lot of opportunities, as Dr. Graves noted, to do more things in the future. I think the innovations we're going to see through stem cell treatments are many. We need to figure out how to repair the myelin that's been damaged in the process. The nerves actually are destroyed in some cases, and there's plenty of work to do. We need to get back and understand what causes the disease. We need to find out how to intercept that earlier and begin treatment earlier. So it's a wide open field. We're in the game. I look at the work that's been done and applaud it and look forward to say there's a lot more to do. And thank you for the opportunity to talk to all of you, and keep up the great work. It's really important that we continue the kind of funding our country has been #1 in the world at for so long. There's some headwinds with that, and we need to figure out a way to get through that. So God bless the work you're doing, and keep it up. And thank you.
Thank you so much, Grant. That was so amazing. We really appreciate your leading the way and talking just not about neural stem cell aging, but also human resilience and the ability to recover and have endogenous regeneration or repair. So you're a living example of that. We do have someone else, Grant, who's joining you as a patient advocate, but more in the Parkinson's disease space. We have Mark and Tracy Ginsburg joining us, and we'll pivot to talk about what it's like to be a caregiver. We heard from Grant really as somebody who's facing the disease himself, and we'll see if Tracy is still on the line. We're a little bit over on the schedule, but it's because we've had such compelling presentations. I think we'll also jump right after that to Nate, our artist, and then we'll go to a brief panel discussion just to introduce the idea that we're all accessible. We all want to be able to ask and answer questions. In other words, we'll ask questions of you too, not just answer. And then we'll be able to see if we can get Mark and Tracy. So Mark and Tracy are patient advocates for Parkinson's disease. We've heard this common theme of aging and inflammation, whether it's how you recover from Dr. Chachi's very precise replacement therapy for spinal cord injury, whether it's Dr. Sharona Benheim's therapy for Parkinson's disease, which is a cellular replacement, or epilepsy, or whether it's Dr. Graves's therapy for MS and also other inflammatory diseases of the brain. So Mark and Tracy have been waiting patiently as we discuss the other things. Here we are. Thank you for joining us. So we just wanted to have you go through what it's like to deal with a diagnosis where the path may not be as obvious or charted in terms of therapies. And we'd like to hear equally from you, Tracy, and from you, Mark, to get your equal perspective, if you want to kick it off.
Well,
I'm the one with Parkinson's. Tracy's a caregiver. MSA, former Parkinson's. I've had it for about 2.5 years. First 2 years were not a big deal, I can say. But in the last 6 months, it's taken its toll to some extent. And it's made it more difficult for Tracy to deal with me. She can tell you the headaches, but I try to keep them to the minimum as best I can.
Mark's MSA, which is a cousin to Parkinson's, as Dr. Jameson had kind of described it, multiple system atrophy, and it is a very unique neurodegenerative disease, and it definitely has its challenges. Mark has difficulty standing, walking, sleeping, has low blood pressure, and obvious Parkinson's isms. So navigating everyday life is very unpredictable. Some days are very good, and some days are not. And some days he has more energy than others, and some days, you know, the movement just isn't there, and we're not quite sure why. There's no rhyme or reason to it, and it's definitely frustrating. I can say as the wife and caregiver, and just watching my husband go through this, it's really difficult. But we are, I guess, cautiously optimistic and hopeful that Dr. Jameson and her team are really doing research to find treatment, and her drug trial that is going to be cutting edge and state of the art. We're hoping that it can really help reduce his inflammation by 50% and his symptoms in his brain. And what else can we tell you? We really appreciate the doctors and the scientists at UCSD, and we put our faith in your hands, and we are really hoping that that we can find a proper treatment and hopefully one day a cure for this disease. And we are really honored to be here tonight, and honored to be patients and patient advocates.
I'm not honored to be a patient. I really want to be a patient.
We don't want to be a patient, but we would like to help find a cure, and at the very least, some really positive treatments for this horrible disease. And we thank Dr. Jameson and your team immensely, and we really hope you keep up the research and find us a cure soon. And we thank you, and God bless you.
Thank you, Martha and Tracy. So what we're talking about there is the inflammatory side of neurodegenerative disorders, and that we may be able to slow that by repurposing or making new drugs that slow that inflammatory basis for neurodegeneration down. And to talk about the precision of diagnosis, as Dr. Graves alluded to, that not everything that it looks like Parkinson's disease is Parkinson's disease. This is multiple system atrophy that Mark is dealing with, and that's what my dad had. So it's very personal to me to be able to take our whole team in this room to find really great, tractable early solutions for Mark to stop this degeneration of his nervous system and to really tackle MSA. We need new ideas. I would like to introduce our artist for this evening, Nate Zoba, who will come here and talk about art for a change. We've got some people, particularly Catherine Canjo, who runs our art gallery here, our really spectacular Museum of Contemporary Art. We started this to talk about the art of science and medicine, but we don't usually talk about how art is done or how people are inspired to do art and be artists and guide us to be a bit more creative. And so Nate, if you don't mind joining us up here, and then we'll have the panelists come up to be peppered with questions.
Okay, good evening. Thank you, Kat, for inviting me to participate in this event. I'm an artist. I live and work in Los Angeles. I have some insight into the world of science and a tiny bit into science itself. IPS is not an uncommon term in my household. I had originally intended to relate this talk vaguely to tonight's scientific subjects. We'll see if I get there. I'm going to talk about a specific body of work, and within that body of work, the specific concept of perception, though the body of work contains many concepts. The 10 paintings and interspersed detail shots being projected behind me Or from that body, and I began it about a year ago. The painting and the details loop twice through the video, and there are 2 paintings here today for you to view in material form. All of this work is oil paint on canvas or linen. My creative academic background is in language, poetry specifically, which I worked in intensely for about a decade. My poetry moved from traditional forms to engagement with the visual aesthetic elements of the page and space, and took on concerns beyond language. Some might refer to it as concrete poetry. In 2011, I began a rapid shift from working with language image material to making work that had no language or symbolic elements in it, primarily working in non-objective abstract painting, which I continue to work in. According to the philosopher Susan Langer, both art and in logic, which carries scientific abstraction to its highest development, abstraction is the recognition of a relational structure or form, apart from the specific thing in which it is exemplified. For me, abstraction is also a way to engage with ideas without using language. Abstraction is a distillation, and distillations are more immediate. Abstraction can contain a multitude of entangled ideas that emerge and recede at different times and from different vantage points. Abstraction can collapse time. But before the abstracting, all of my work is rooted in observation. My observation tends to be less focused on single objects or settings or landscapes, and more focused on my reality, which could be thought of as my mind. I observe the cumulative experience of the present or specific reality is accessed by me through my perceptions, and attempt to observe the perceiving as well, which I then distilled into these paintings. These paintings engage with the myriad perceptions and observations of the last year and the way I was perceiving. And there was a lot to perceive, especially in Los Angeles. From moving painting studios to days of wildfire smoke, smell, and darkened skies, radically changing power structures and forms of government, to the city's protests of new assertions of control and its effects, and of course the many other global events delivered to me digitally. I became hyper-aware of how I was perceiving the perceptual effects, the constantly shifting between the proximate phone and its ever-varying virtual depth and its backlit hyperkinetic activity, with the larger visual field of the physical world with its steady depth and semi-regular motion, the elm and the grass of the backyard, the billowing sunsets, observing time and light through sidewalk shadows. I considered how the brain constantly shifts between these radically different spaces, from the depth of the world to the screen surface to virtual depth of that surface, its content, and back to the world depth again, over and over. How the digital records a form of the physical, and in the physical world, the recording digital is always present. How the aesthetic qualities of these spaces collapse into and overlap each other, creating a non-existent third space that is very real to us. How digital spaces have a directional motion in scrolling and swiping compared to the steadier and consistent motion of the physical world. In the mind, there becomes a space that has qualities of both the digital virtual and the organic physical, but is not wholly one or the other, is not located physically in the world and not virtually in the screen, only in the imaginal spaces of the mind, but not purely imaginative nor purely perceptival. It is a simultaneous space our brains have never really held, but now hold for much of waking life. To me, the additive and excavatory marks in these paintings are the mind's aesthetic response to this synthesis of the virtual physical. Its attempt to hold together, to capture motion, information of reality, and its attempts to break it apart is being unnatural and unknown, being in this dual position that is so true for much of our lives. The absorption of the frenetic activity of the incessant backlit digital spaces, its flickering, notifying, interrupting, connecting, communicating, reminding, pseudo-remembering, the constant motion of saturated color in a field of virtual depth. It's always juxtaposed to the geometric and organic regularities of the physical world that extends steadily and regularly in all directions out from the screen, in which the mind has long known and is most familiar. I wanted to make paintings that look like how that space feels and have that feeling be a reflection of the space in my imaginal reality. An abstraction of reality held in the imagination, but real, a perception of our perception, and a connection to the space in which we exist, but do not yet name. Thanks.
So thank you, Nate Zoba. That was spectacular. So that's the art and poetry of perception and cognition. And it seems somewhat mathematical, I've got to say. Nate, I think you are a scientist of art, though. So I'd like to invite Jennifer Graves, Sharona Benheim, and Dr. Chachi to come up here. I know what we've discussed today looks improbable, maybe even impossible, but it's already being done. So what we really need your help with now is the future. So come on up, and if you take a chair here, I'm just going to let Dr. Graves, Dr. Benheim, Dr. Chachi answer your questions, because I'm sure you have many questions and would like to ask them. Anybody's free to ask questions. We have people from the Sanford Stem Cell Clinical Center, Dr. Morris back there. So he'll answer all your questions about logistics for implementation on some of the clinical trial aspects of things. But, you know, there are some obvious things to think about here. You were a dancer before, Dr. Graves. You clearly have a lot of energy. What propels you to persist in a pretty tough disease setting, progressive MS? Why do you're a powerhouse in and of yourself to do all of this? What compels you to do that?
Well, thank you for asking that. I was a dancer when I was younger, and when I was looking for what I wanted to do in science, it's actually part of why I became an MS doctor. I saw a disease that was hitting people very early in life, removing their ability to move, and it actually was a big motivation. And in fact, on top of all the other things we do, pushing forward on the medical side, we've actually started a dance program for people living with MS, as well as some arts programs, because that complexity of art that we just heard does help train the mind and help preserve it. But it is an amazing population. I don't know if I shared this with you. I might have shared it with someone else, but I just finished a 50-mile walk for MS. Yikes. And I did it with people living with MS, so by my side. So it's an amazing population to work with. There's 1 million people in the United States facing MS. My kids with MS face 70, 80 years with this disease. They're my biggest motivation. And they provide insights. I think all of us, we see certain nuances of the disease in the people we take care of, which leads to new ideas.
Great, thank you so much. Grant, we'll have you on the spot too for questions in case anybody have them, but I wanted to ask Dr. Benheim, so you're a surgeon that's looking at these adaptable pathways when it comes to epilepsy. I've got to say, when we considered the Neurona trial in terms of providing our support for that, I said that will never work. It's just too complicated. There's so many moving parts. What made you think that it would? I mean, having had spectacular outcomes, and the same for the Parkinson's disease trials, plural, that are stem cell related trials.
Yeah, thanks for that question. So epilepsy is really complicated, and there's so much about epilepsy that we actually don't yet understand. And then to make it even more complicated, the disease varies significantly from person to person in how their seizures manifest, where their seizures start, and how they spread. And then to make that even more complicated, that changes with time even more. But right off the bat, I was very excited about the concepts relating to cell therapy for epilepsy, because not only do we have a tractable target for us to treat now, we also have a vision of how to treat epilepsy for the future that might actually be more commensurate with how the disease actually evolves and progresses. And so just to kind of give you an example of that, right now we conceive of seizures in patients who have a specific kind of epilepsy, which we call focal epilepsy, as starting from one part of the brain. it may then secondarily generalize and evolve from that part of the brain to involve the whole brain, but the concept is that it starts in one part of the brain. And so this trial and this therapy allows us, after we have identified that part of the brain, which in this particular trial is the hippocampus, the most common part in adult onset epilepsy, this trial allows us to now inject cells that are going to cause inhibition to that part of the brain, and by doing that, ideally restore the balance between excitation and inhibition that we see very much go awry in epilepsy, and we have some good data for that. But what's really exciting about it is that we're coming to understand that epilepsy is not just a focal disorder, but a network disorder. And probably it's really not just that one part of the brain, but various parts of the brain that are interconnected that actually cumulatively affect patients with epilepsy. And so the way we're thinking about it is kind of simplistic, but we have to think about it this way because that's how those are the options we currently have to treat patients with epilepsy. Cell therapy opens up a whole new variety of options because now that we don't have to go in and resect or ablate those parts of the brain, and we can actually augment the way they function, we can really potentially treat epilepsy in the form that it actually really exists. And so it is really exciting, not just for the present day of epilepsy treatment, but how we're going to both think about and treat the disease in the future.
Yeah, and you've got one patient I know of who's been here for a mind event that went from having 30 seizures a month to 2. So he's 2 years out now. Spectacular work. So Professor Chachi, you got us all started on this. You're an innovation pioneer. You develop new techniques, tools. What is our inherent responsibility to drive the field forward? How do you feel about surgeons, scientists, innovators being involved in the development of these technologies, particularly with the DOSE study where you developed a better way? I mean, again, 0 adverse events with Dr. Chachi's neural stem study. I anticipate it's going to be the same thing with the next oligodendrocyte progenitor. What's the role for a surgeon, scientist, and leader in the field like you to develop the new technologies?
Well, we have a great responsibility to do so. We are gifted in our profession to have access to the central nervous system and the peripheral nervous system. And so we have a great responsibility to use that access to try to deliver therapies and envision therapies and create mechanisms to get those therapies into the patients that need them. And I also rely a lot on brute force and ignorance.
So that self-deprecation is pretty funny. When I heard the last time, I thought, Italian, no. Dr. Chachi's an Anglophile. He loves Doctor Who, which is why he shows the phone booth at the end. So you can see everybody is a Renaissance person here. They've all got multiple talents and certainly very gifted. It is invented here. It's invented by the people you see in front of you. The technologies, the cellular therapies that we're developing, we want to administer them to the right person at the right time. But just wanted to open up to questions, perhaps from an editor who published this paper. Not to put Sheila on the spot.
Well,
she's
I have
just
some
here to listen.
questions.
We have questions still for Sheila. For Dr. Chari, as a lead editor for Cell, one of the top journals, Cell Stem Cell, where you're leading in terms of allowing us to have a public voice and platform to be able to show why this matters and in whom. What are your thoughts about the future for this field? I mean, you've got all of our friends here, patients on the line, no pressure. How should we frame this so we convey the messages accurately?
Well, I'm very excited to see all the range of diseases that you're starting to treat now, and it's interesting to see. What excites me the most is how safe all of these cell therapies have turned out to be. Obviously, you're selecting and working with patients that have disease that perhaps, as you said, they're volunteering and they aren't necessarily expecting, curative effects. But I wonder if you could all talk a bit more about the safety profile of cell therapies and what you see for the future.
Maybe Dr. Chachi first, because he's always going to let everybody else go first. But Dr. Chachi has the longest experience with safety, these trials.
Correct. And, it's really a matter of refining the technique before enrolling any patients. And so we, in preparation for every clinical trial that we've done, have done a lot of training and a lot of dry runs, sort of drilling is what we would call it. We have regular drills and really go through the process at every level, not just the surgical technique, which is super important, but from loading cells, every other aspect of it. And then, got to give credit to our ORs and our support staff in the ORs who do a really good job taking care of complex patients all the time. I mean, just Dr. Beaumont's complications alone would really train people up for crazy scenario. Sorry, Tom, but I'm glad you're here. Thank you. No, it's really about mental and physical preparation. Surgery is a mental thing, but it's also a physical thing, and really requires getting your technique down and also having a plan ahead of time. It's not an ad hoc situation. We really did think about this a lot, and you need really brilliant collaborators who have really gentle hands.
Uh-huh. Yeah, so we do have the Sanford Surgical Training Center. So Dr. Chachi has insisted on training before every surgery with his entire team. So cerebellar motor memory, yes, but it's also team dynamics. So well done. Thank you. So Dr. Benheim.
Spine surgeons can have gentle hands too. But what I didn't really actually touch upon too much in my talk, but I think is worth noting, is that just how minimally invasive some of these therapies can be. In these trial forms, that's not necessarily our focus in all of them per se, but what we're doing here is we're using a needle that's typically somewhere between 1 and roughly 1.6 millimeters in diameter to actually go into a target structure in the brain and inject these cells. And so we can do that really with just a tiny skin incision and a very small opening in the skull. That's roughly usually 2.4 millimeters, and it's a one-stitch closure. So of course there are always going to be inherent risks in doing any sort of surgery related to the brain. But our techniques and our ability to visualize key structures in the brain, our ability to use tools like real-time MRI imaging, like robotics, allow us to get really sub-millimeter precision in what we're doing that allows us to do this safely now, whereas in the past, these types of techniques and technologies weren't necessarily readily available to us. So it really is a whole new era of safety.
And Dr. Graves, you're dealing with cells that find their own way. So CAR T cells in an inflammatory disorder where they can cause inflammation. How do you mitigate the risk?
Yeah, so it's a great question. So far, so good. The CAR T cells that have been delivered to people with multiple sclerosis, we're at the beginning stages. We've got multiple products rolling out into clinical trials. We're watching very closely. And I think it just highlights how important it is for this to be a team-based sport. So we have the neurologists working with some of the oncologists that have more experience with the CAR T. We're all braced and ready for looking at things and monitoring things. But so far, so good. But it is an open question. When you take immune cells and you put them in someone with clear immune dysregulation and disorders, it's a risk. But it's a risk that could have huge payoff. Mm-hmm. So it's why we have to be willing to do bold science. Bold science, requires partners that want to do bold science because, we have to push the frontier, team-based sport, and people willing to fund this work they're willing to take risks. But so far, it's actually been much less side effects than we were even anticipating. So far, it's working out great.
I know we have the Sanford Stem Cell Clinical Center Cell and Regenerative Medicine Service to help to consult if there's an issue. But I wanted to thank you so much to the three of you. I wanted to give the last word to Grant because you have held on valiantly here. If you would like to give us the last thought about what we should be doing next, or what you think, what you're hoping for the future from our august team here.
I think the notion of trying to really understand the blood-brain barrier and thinking about what can be done to make it a more challenging environment for these nefarious cells to kind of leak into the brain would be a great thing. And I think there's a lot of tissue repair. I mean, ultimately, you know, you could imagine a day where destroyed nerves can be regenerated and full function restored into patients who have lost, you know, mobility, lost control of a lot of functions. I mean, that'd be a great outcome down the road. And the kinds of technologies required to do that, some are in their nascent form today, in all likelihood, but I would expect if we had this sort of conversation in another 10 years, it'd be a whole different dialogue. So it's an exciting time, and you got to push the frontiers.
So you always have the person that is actually dealing with the disease to come up with the most brilliant ideas. Thank you so much, Grant. That was incredibly helpful. I'd like to thank the audience for taking part in this. We'll be back for another Mind event coming up very soon. Thank you.